Now we are going to learn about the complement system. The complement system consists of a number of proteins. We know that our human serum contains different types of proteins. The complement proteins are one of them. These are the proteins which are normally found in inactive form. Generally, they do not have any function, but once they get activated, they cause augmentation or the enhancement of the immune response, which is already activated in the human body against a particular pathogen or the antigen. So how can we define the complement protein? See here, what I have written is that the complement refers to a group of the plasma proteins which are normally found in inactive form, and they augment the immune response. They augment the immune response when activated. So this is the proper definition of the complement proteins, that they are the normal plasma proteins which are present in inactive forms in normal situations, but when they get activated they cause enhancement or the augmentation of the immune responses against a particular pathogen or the antigen. That's the definition of the complement proteins. Now in certain universities, they have been asking about the what are the properties of the complement proteins. So here I have enlisted certain properties of the complement proteins. Let's see one by one. The first property of the complement proteins is that they are proteinous in nature. Okay, this is quite obvious from their name as well, that they are proteinous in nature. Then the second property is that the complement protein's serum level does not change by any infection or any vaccination. It is not like the antibodies levels, like the antibodies level change after vaccination or after certain kinds of infections, but the complement protein level does not change even after any vaccination or the infection. Their serum level remains constant. That is the very important property of the complement proteins. Then the third important property of the complement proteins is that they are species non-specific. What does that mean? So the meaning of species non-specific is that they are present in all the mammals and the birds, amphibians, fish, etc. They are not only limited to the humans, rather they are present in all the mammals, birds, amphibians, etc. Okay, so that is the meaning of that, they are species non-specific. Okay, then the complement proteins are heat labile. The fourth important property is that the complement proteins are heat labile. What does that mean? This means that the complement proteins get denatured at 56 degrees centigrade if they are heated for 30 minutes, and if serum is heated at 56 degrees centigrade for 30 minutes, that means all the complement proteins in that serum will get inactivated, and that serum will be called as the inactivated serum. So that's the definition of the inactivated serum as well. Okay, so a serum which contains inactivated complement proteins, okay, or denatured complement proteins, that serum will be called as the inactivated serum. Now the fifth property of the complement proteins is that all the complement proteins has two fragments. The larger fragments is referred to as B fragment, and the smaller fragment is referred to as the A fragment. Generally it is seen that the larger fragment is B fragment and the smaller is A fragment, but in case of complement protein 2, the A fragment is bigger than the B fragment of the complement protein 2. So that is a exception. So see here, I have here mentioned also that the larger fragment is B and smaller fragment is A fragment of all the complement proteins except the 2CA. Oh, sorry, the C2A, that means the complement protein 2A, that is the bigger fragment than the complement protein C2B. Okay, the next property of the complement proteins is that these complement proteins are activated by proteolysis. You see that whenever a complement is broken, then what happens, that the complement proteins are broken into the A fragment and the B fragment. Now this breaking of the complement proteins into the A fragment and the B fragment, that is happening by what method? The protein lysis always occurs by proteolysis now, so that's why the complement activation occurs by the proteolysis. Whenever these complement proteins are broken down into the A fragment and the B fragment, then the activated the binding site over the large fragment gets exposed after the removal of the smaller fragments from that site, and then the complement proteins get activated, or the larger fragment of the complement proteins get activated. So that's the meaning of activation of the complement proteins by the proteolysis, that once the smaller fragments are removed from the larger fragments, then the binding site over that larger fragments get exposed, to which the antigen antibody complex can bind or the antigen can bind, and thereby the complement system can get activated. Okay, so that's how the complements get activated by the proteolysis. Then the seventh and one of the important property of complement proteins is that they are synthesized in the liver. So where does the synthesis of complement proteins occur? It occurs in the liver. Now we are going to learn about the complement system, the categories, the classification in the complement system. Okay, so the complement system contains about or consists of about 30 plasma proteins. Now all those 30 plasma proteins have been classified into three groups. Okay, and that groups are the complement component, then the protein system, and the regulatory proteins. The complement components contains all the complement proteins which are numbered from C1 to C9. So there are total 9, the complement proteins, specific 9 complement proteins which are numbered from C1 to C9, which are taking part in the complement activation, okay, or the complement immune response. Then we have the protein system. The protein system, for example, the protein that is included in the protein system is the factor B, which is required in the alternate pathway of the complement activation. We will see there are different methods of complement activation, one of which is the alternate pathway, and in the alternate pathway we require factor B for the activation of the alternate pathway, and that factor B comes under the protein system. So this protein system is also a part of the complement system. Then we have regulatory proteins also for the regulation of the complement system, the example of which is the C1 inhibitor protein. Now this C1 inhibitor protein, what does it do? This C1 inhibitor protein breaks down the C1 complement protein into C1q and the C1rs components, so that the classical pathway of the complement system activation could not occur, and thereby it inhibits the classical pathway of the complement activation. So thereby it regulates the complement system. So these are all together comes under the these are all together complement proteins which come under the complement system. And then we have the complement activation. So the complement activation can occur in three ways. There are three pathways of the complement activation. The first one is the classical pathway, then we have alternate pathway, and then we have lectin pathway or the mannose lectin pathway. The classical pathway is activated by the antigen antibody complex, while the alternate pathway gets activated by the antigens. Okay, the antigen triggers the alternate pathway activation. This includes the bacterial endotoxins, then yeast cells, viral antigens, etc. Then we have the mannose lectin pathway or the lactin pathway, that is activated by the mannose residues on the microbial surfaces. So we will see one by one all these pathway in our next videos. That's all for the first part of the complement proteins. Next we will see the activation and the different pathways of the complement activation.